Hello, my name is Wesley Ray, and I am a student in the master's program of biology at Grand Canyon University. And today I am going to be recording the lecture for what's my last assignment in my Biochemistry 530 class and the last assignment for my master's overall. So I'm happy about that. Uh, but without any further ado, I'll go ahead and I'll share screen. Um, I apologize if things are dark in my room. Um, Again, it's kind of late when I'm working on this, so um, but let's get to it. So I'm going to share screen and bring up my PowerPoint presentation. Get that shared. I'll show from the beginning. All right. So today we're going to be talking about glycolysis, gluconeogenesis, and the role of aldolase gene expression. Oh my goodness. Oh, up now. Let's start over. So we're going to define by breaking this down into four different kind of subtopics. We're going to give an overview on glycolysis and gluconeogenesis themselves, what those processes are and what they entail, why they are important, and how they are used in the body. We're going to talk about what aldolase does as an enzyme for the processes of glycolysis and gluconeogenesis. We're going to talk about the different isoforms of aldolase and where they are expressed in the body. That was my bird. And then we're going to talk about some specific transcription factors and different binding proteins and what effects they potentially can have on the expression of one of those isoforms of aldolase, aldolase B. So, starting off with glycolysis. Glycolysis is a very, very old ancient metabolic pathway. It predates oxidative processes. So, this is what bacteria were doing in a time back before there was a significant proportion of atmospheric oxygen. Okay, oxidative phosphorylation was not a thing, and already glycolysis was old news. This is the original cellular metabolism as we understand it. Okay, it focuses on starting with glucose or a simple sugar and breaking this down into pyruvate and other different metabolites. Okay. Um, it does produce ATP as a portion of this reaction. I believe it produces 2 ATP. So compared to something like oxidative phosphorylation, it's a very inefficient means of producing cellular energy. But in the absence of oxygen, it's what you've got. So this is allowed to proceed because of a chain of enzyme catalyzed reactions. Okay. This process normally would be so slow that it would be non-functioning for any living life form as opposed to dead ones. But this chain of reactions has got to be catalyzed. So you've got a whole slew of enzymes that are produced by cells that sort of allow this process to move forward. If you were to disrupt the expression of any of these enzymes, you will disrupt the ability of a cell to go through glycolysis. So all of these enzymes are vitally important. Okay. It's an anaerobic process. I kind of mentioned that. It still is used by any number of anaerobes today. I mean, if you go down into soil and you go down past that first 6 to 8 inches, you're going to find a whole slew of bacteria that still use this process almost exclusively for their cellular metabolism. Us, as kind of more complex eukaryotic aerobes, we still use this as sort of the first of three sets of reactions in our cellular metabolism. Of course, we finish that off with the electron transport chain, oxidative phosphorylation. But again, in a time before that, this was it. The enzymes are encoded in various different locations, and the expression of them has regulatory implications. Um, Different aldolase expressions actually have relationships with different canceral lines. That's the article that I cited there, I believe, talks about aldolase A with various cancers. Um, One of the things about cancer is that it has sort of a disrupted metabolism because of a phenomenon called the Warburg effect. And that sort of means that they lose the ability to go through oxidative phosphorylation and they depend exclusively on the metabolization of glucose, meaning that cancer cell metabolism is oftentimes disproportionately dependent on these enzymes for their sources of energy. So, fun little factoid there. I have the image here. I just to kind of explain, and I've sourced my link below it. But this is the process of glycolysis broken down by the specific molecules and their respective enzymes to go in between them. I will note of particular importance that if you look in the top right of this diagram, you see the moment that we're going from a six carbon chain to a pair of three carbon chain containing molecules. That's going to come up later. We can contrast this with gluconeogenesis, which is essentially, oh, hey, Edward, which is essentially a reverse reaction or a reverse set of reactions to glycolysis in the first place. It is a metabolic pathway that uses enzymes to convert metabolites into glucose. So we're going in the opposite direction. Instead of glucose into pyruvate, we're going from pyruvate into glucose. The enzymes are going to differ a little bit from organism to organism. Scientists that have studied this have tried to use the genes as a way of trying to map the evolution of some life forms. This was first studied and laid out in dogs by a scientist, I believe, by the name of Claude Bernard. Um, but it's been observed. One of the chronologically organisms that were supposed to have first developed it were cnidarians, worms. So something to think about there. It's induced by hormonal secretions of the liver to regulate blood glucose levels in humans. So yeah, this whole thing, gluconeogenesis versus glycolysis, is sort of the story of glucagon versus insulin manifest, the effects of them. Um, Insulin is sort of pushing forward the process of glycolysis, and uh, glucagon pushing forward this process of gluconeogenesis. And in our bodies, we use this as a regulatory mechanism for blood sugar. That's not necessarily universal, but we can talk about it in human terms. I will say that because this is a chain of reverse reactions to glycolysis, you will see many of the same enzymes. And aldolase that we talk about later is one of those enzymes that is apparent in both glycolysis and gluconeogenesis. So, yeah, be on the lookout for that. I'm going to call that a decent overview of glycolysis and gluconeogenesis. And that should let us proceed to the role of aldolase, respectively. So I have aldolase, human muscle aldolase. That should be aldolase A. That should be aldolase A if it's human muscle aldolase. Aldolase A being expressed in those areas and in blood cells. Um, yeah, it's an enzyme present to catalyze the fourth reaction in glycolysis. Okay, so I've brought in that same picture that I showed you earlier, and I've sort of put a box around the area that I mentioned. Okay, this diagram brings up fructose biphosphate aldose. This is where aldolase is acting. Okay, this is where all the research studies have pointed me towards that this is where aldolase is working. Aldolase is the enzyme that is breaking this fructose 1,6 biphosphate down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And this is a reversible reaction. We can go between these two depending on gluconeogenesis or glycolysis, whichever pathway you are going down. Okay. And yes, as I mentioned prior, aldolases are responsible from going from that six carbon chain to the three carbon chain. You'll notice in everything to the left of the box, we're in six carbon chains post the glucose. Once we get out of that sort of cyclohexane structure, and we go to a more linear, sort of more linear structure. Yeah, it's the six carbon chains being broken down into pairs of three carbon chains. Okay, the reactions are reversible. In gluconeogenesis, aldolase is working in the same place. It again is just a difference in the direction that we're going in the reaction. Are we starting at pyruvate down here in the bottom left and going up to glucose? Or are we starting at glucose and going down to pyruvate? Okay. And if you disrupt aldolase expression, you're going to disrupt both of these processes. They will not proceed without expression of aldolase. Okay. There are, again, possible cancer implications due to the presence of the Warburg effect. Okay. Isoforms of aldolase and their expression patterns. There are three characterized aldolase isoforms that are discussed in the terms of humans, and also all mammals from the information that I could find. Several of the articles that I cite are built around studies where they are testing different, knocking out this gene and seeing what it does to expression levels. That's being done in rats, in rat hepatocyte cells. But the implications of this are mammalia wide, I'm going to say. Aldolase A seems to be expressed in muscle and red blood cells. I kind of mentioned that earlier, and I hope I got which one of those it was right. I'm not working off of notes. Aldolase B is expressed in, I'm going to say that this is sort of expressed in the smooth muscle tissues. Seems to be the trend. It's being expressed in liver and hepatocytes, the term for the liver cells. It's being expressed in kidneys. It's being expressed in intestinal tissue. And aldolase C being expressed in nervous system tissue and the central nervous system, the brain in particular. Towards sort of the latter part of this presentation, we start talking about the implications of various transcription factors on aldolase B specifically. I want to draw attention to that there are different modulators to aldolase A and aldolase C. Okay, they are very similar in size. They are very similar in length of gene, and they are very similar in role. They do the same job. It just depends on where they're doing it. And they have sort of a difference in preference to which direction they want to go and what they want to work with. You've got some that would rather work with glucose and some that would rather work with fructose. You've got some that want to move forward in the reaction sort of glycolytically. And then you've got some that want to move backwards, gluconeogenesis. They all can be used both in glycolysis and gluconeogenesis, but they have sort of their innate preferences to moving forward or backward if we're talking about glycolysis as a reversible chain of reactions. Okay, so the major difference is where we're talking about. All right, HNF1, HNF3, CEPB, and DBP, which is D-binding protein, expression on aldolase B. So I have aldolase B shown there pictorially. These diagrams, and again, I've given the link that I've sourced that from. These diagrams are made in softwares. This is either made in or made in something similar to UCSF Chimera, which is a cool software for those who haven't used it. You can go into different databases and find these codes that you can input into UCSF Chimera, and you can sort of model different proteins. You can color different structures based on chemical characteristics, composition, charge structures, all sorts of different things. Secondary structures, you can see alpha helices very plainly labeled there, etc. Aldolase B, predominantly expressed in smooth muscle organs. Okay, it shows no noticeable preference for the direction of reaction. So, in tissues that we're looking at, usually in vitro, there seems to be no preference for aldolase as far as its participation in glycolysis or gluconeogenesis, specifically aldolase B. It is negatively related to insulin expression in humans, apparently, which I think is interesting. So, HNF1, this is hepatocyte nuclear factor 1. Hepatocyte being liver cells. Okay, so Hepatocyte nuclear factor 1 is a transcription factor acting as a strong promoter of aldolase B expression. If you overexpress HNF1, you're going to overexpress aldolase B. If you underexpress HNF1, typically the expression of aldolase B will decline. You have multiple different transcription factors that are acting as promoters. So just because if you were to say have a mutation, or if you were to do a knockout of HNF1, it doesn't necessarily mean that you won't express aldolase B, but the expression will typically trend downward. Okay? They are positively related. Less of one means less of the other. More of one means more of the other. We can contrast that a little bit through HNF3, which is hepatocyte nuclear factor 3. The naming schemes are highly original. An alleged repressor of aldolase B expression. So this is negatively related in laboratory studies, and the hypothetical mechanism here is that it's actually a promoter. It's a weak promoter. And HNF3 and HNF1 are competing for the ability to serve as transcriptional promoters for aldolase B. If HNF1 wins that competition, you're going to have an increased expression of aldolase B. But because HNF3 is such a weaker promoter, if HNF3 is overexpressed, you'll begin underexpressing aldolase B. It is still a promoter, but when compared to HNF1, it's not a very good one. It's not a very strong one. And we use HNF3 apparently as a regulatory mechanism to sort of downregulate HNF1 or downregulate aldolase B as needed. So kind of a weird case of competitive promoting acting as an inhibitory mechanism, if that makes sense. CEPB, that's C-cat enhancer binding protein. This is a promoter of aldolase B expression. Regionally, it is more or less important when looking at liver tissues versus kidney tissues. It's most important in liver tissues. It is less important in kidney tissues. I am unclear as to why that is, just to be completely and totally honest with you. But there are patterns of expression that you're going to see more of this being expressed in hepatocyte tissues more than you're going to see them in kidney tissues. Okay, and we're looking at that studies by Gregory. This is Claudine Gregory. Is cited in my presentation here. I cite maybe 4 or 5 different research articles that are all coming from that person or teams that they're leading. I guess this person is like the aldolase B expert, and they do a lot of different studies in rat hepatocyte tissue. And they conducted a study where they were doing I believe they were knocking out the C-cat enhancer binding protein in these rat tissues, and they observed less of an effect on aldolase B expression in kidneys than what they observed in liver tissues. DBP, D-binding protein. This is acting as a promoter of both albumin and of aldolase B in human hepatic cells. Very important. However, when you take away DBP, it seems like it doesn't have quite as large of an effect as what the CEBP or HNF factors seem to have on aldolase B expression. Okay, and I have my references. So with this being the last project, I just wanted to give kind of a brief shout out and thanks to our professor, Dr. John Wool. I've appreciated the class, and to Grand Canyon, I've appreciated the program overall, and I hope you guys enjoy the presentation. Thank you. I'm going to stop my screen sharing, and I'll stop recording.